Hello, this is Dr. Bates, and we're talking about Chapter 13, Principles of Disease in Epidemiology. A patient entered the hospital to have a torn cartilage removed from her right knee. The surgery was scheduled as a same-day procedure, should be in and out. Unfortunately, she subsequently developed pneumonia and wasn't released until 10 days later. How would you account for these events? Accounting for these events actually fits into epidemiology. So we're going to talk about terminology, microbiota. We're going to talk about Koch's postulates again. We've mentioned them before. We're going to talk about disease incidents and descriptions, and we're going to talk about emerging infectious disease. So first, some terms. Pathology is the study of disease. Etiology is the study of the cause of a disease. Pathogenesis is the development of a disease, and those would be caused by pathogens. Infections are where we have a colonization of the body by those pathogens. And then the definition of disease is an abnormal state in which the body is not functioning normally. Now, there is some argument about this definition of disease. There are some that believe that a disease is only something in which a infection is causing the abnormal state. Therefore, something like heart disease is not actually a disease, it's a disorder. When we talk about microbiota, we can talk about transient microbiota and normal microbiota. Transient microbiota are only there for a short period of time. That could be up to months. But your normal microbiota is what you permanently have all the time and doesn't change. Symbiosis describes the relationship between us and our normal microbiota. The Human Microbiome Project is a project that's working to see how microbes affect humans and human health. They're also trying to categorize what microbes we actually find interacting with humans, and that diversity. Here's some examples of normal microbiota. Here we have bacteria, that's these orange spheres, and these guys are on the surface of the nasal epithelium. Here we have some bacteria on the lining of the stomach, and here we have some bacteria in the large intestines. The types of microorganisms that you have as your microbiota and where they're located is determined by the nutrients, physical and chemical factors, the host defenses, and mechanical factors. When we talk about symbiosis, we can have commensalism, with one organism benefits and the other's unaffected, mutualism, where both organisms benefit, or parasitism, when one organism benefits at the expense of the other. There's some argument whether commensalism actually is a real thing. Can you think of an instance where one thing would benefit and it would not affect the other item? This was originally thought when we talked about microorganisms in the concept that staph epi on your skin would be a commensal. It just lives there and it gets the benefit of a place to live, but you don't get any benefit from it. However, with the current knowledge, it is known that it is actually providing a benefit to you. Therefore, it's not really a commensal. It would be mutualism. Now, some normal microbiota, organisms that you find on humans all the time, are also opportunistic pathogens. That means that if the opportunity arises and the immune system is compromised, that they might actually start invading tissues. Now, we don't find microorganisms inside your tissues unless you're ill or they've been invaded, and that's a bad thing. We find them on anything that we consider to be external. So your skin, of course, your digestive tract, etc. Those are open to the environment, and that's where we find microbes. So here's just examples of normal microbiota and what we found. So here we have the skin and a list of microorganisms. Notice it includes bacteria and fungi. Your eyes also have microorganisms. The microbes on your skin have to be able to handle the environment that's in your skin, and that is that you have low pH, that you have secretions that cause that environment to be antimicrobial. So only some organisms can live there. Also, there's a low water availability. Your eyes, the conjunctiva is going to have some of the same microbiota we find on the skin. And your tears and blinking actually help eliminate microbes and inhibit other ones from being there. In your nose and throat, your upper respiratory system, we find microorganisms. They could be potential pathogens, but you have microbial antagonism where you have more than one microbe there fighting for a space. Therefore, it makes it difficult for disease to occur. And you have nasal secretions, your mucus and ciliary action help remove microbes. You have microbes in your mouth. It's a moisture-rich, warm environment. But you move your mouth, you chew, you swallow, all of those things are going to help remove those microbes and move them into a more inhospitable environment. Your saliva also contains antimicrobial substances. Large intestine, this is where we're going to find the largest number of resident microbiota because of its environmental conditions. But again, we have things that help protect us from those microbes. And then our urinary and reproductive systems, again, we're going to find a resident population. The environment there is set up to help reduce the number of microbes and especially to take care of ones that might cause disease. So, microbial antagonism, I said that a moment ago. Microbial antagonism, I call this a crowded beach. This is the concept that if you have microbes already colonizing something, then there's not going to be enough space for new ones to take up a spot. If you go out to a beach and every part of the beach is covered by people, you're probably going to turn around and leave. And that's what happens here. Microbial antagonism, the competition between microbes for space, for food, for environmental conditions is going to be high, and therefore that's going to protect you. So normal microbiota protect you by occupying spaces that pathogens could be there. They also can produce substances that can be harmful to other microbes. They affect the pH and available oxygen, again, making an environment for them to keep out everybody else. Now, you've probably heard of probiotics. Those are live microbes that are either applied or ingested that are there to add or influence your organisms that are there by applying new ones. So now that we've talked about normal microbiota, Koch's postulates should make a little bit more sense. If I isolate organisms from someone, I'm going to get quite a few different kinds. So we need to make sure that we figure out which one is the pathogen. So the steps are, the same pathogen must be present in every case of the disease. The pathogen must be isolated from the diseased host and grown in pure culture. The pathogen from the culture must cause disease when it's inoculated into a healthy, susceptible organism. The pathogen then must be isolated from that organism and shown to be the original pathogen. Here's a picture showing that process. So we have microorganisms isolated from a diseased or dead animal. We're going to observe them and grow them in pure culture. Then we're going to inoculate a healthy individual, make sure that it has the same signs and symptoms, the disease is reproduced. Then we're going to again grow them from that organism, verify that it's the same one that we isolated in the original event, again looking at in the microscope, again verifying that they are the same organism. Now there are some problems with Koch's postulates, and that is that some pathogens cause several disease conditions, and it does change based on different individuals. Also, some pathogens only cause disease in humans, so I can't test it in a lab animal. Some pathogens, we don't know how to grow them in pure culture. So until we understand them, we might not be able to actually culture them. When we talk about infectious diseases and we're trying to classify them, we're going to be looking for signs and symptoms. Signs are changes in body function that's felt. These are the hardest ones because you have no way of verifying. The individual says, I don't feel good. Well, what does that really mean? Signs are things that can be measured or observed, such as fever, change in heart rate, things that you can actually measure that are caused by the disease. Now, the syndrome is the specific group of signs and symptoms that accompany a particular disease. Communicatable diseases are, of course, diseases that can be spread from one host to another, whereas a contagious one is that's easily spread. HIV is a communicatable disease, but it's not contagious. You can spread HIV from one host to another, but it isn't easily spread because it requires blood-to-blood contact. Non-communicable disease is something that can't be transmitted from one host to another. If we talk about the occurrence of a disease, we have terms for that. We have the term incidence, which is the fraction of the population that actually contracts a disease during a specific time, whereas prevalence is everyone who has that disease at that given time. So incidence is the new infections. Prevalence is the total. Sporadic disease is something that we see occasionally. An endemic is something that is constantly present. It is found in that area. An epidemic disease is a disease that's acquired by many hosts in a given area in a short time, and then a pandemic is a worldwide epidemic. Now, one thing that can help us with the occurrence of disease is herd immunity. And herd immunity is where a population as a whole is immune to the disease. This is going to stop the spread of the disease. And here's an example. If we look at the top picture, we have a disease where people who are not immunized, no one has a immunity to it. When they come into contact with someone who has a contagious disease, that disease is going to spread very quickly. Now, if we look at another option where some of the population is immunized, that's going to decrease the spread. But if I have a population where everybody but a few individuals, these people are blue, are actually immunized, I'm going to decrease the incidence because it's going to be difficult for that person who has the disease to come into contact with someone who doesn't, because all the people in between aren't going to spread the disease. Back before not getting immunized was unpopular, that was actually an okay thing to do because everybody else getting immunized was actually going to protect those people who don't. Now, immunization doesn't just mean that I went and got the shot. There are other ways to be immunized in that there are individuals who come into contact with a childhood disease that actually ends up protecting them from getting it as an adult. Here is a chart showing reported AIDS cases. I want you to notice this huge jump in AIDS cases. When you look at this graph, your question would be, what happened there? Why did we suddenly have a huge jump in cases? And the answer is, that's when they actually started asking more questions about HIV. It wasn't actually an increase in incidence, it was an increase in reports. Now, I want you to notice that there was an increase and then you see a decrease over time, but that the number of cases stays fairly stable. If you want to read more about AIDS in the United States, you can check out the CDC report about it. Diseases will have a severity or duration. We can have acute diseases, chronic, subacute, and latent. Acute diseases are something that come on rapidly and then go away very quickly. Chronic diseases are something that takes time, builds up over time, gets worse. Subacute is somewhere in between acute and chronic. Latent diseases are diseases that have a period of no symptoms. And during that time of no symptoms, the causal agent is inactive. You might be tested for it and you wouldn't test positive even though you were a carrier. Eventually something happens and the causal agent will probably get activated and then the symptoms will occur. Local, systemic, and focal infections all describe how far that infection is in an individual. So local is where the pathogens are on a small spot. Well, it doesn't necessarily have to be small, but they're not throughout the body. Systemic is throughout the body. Focal infection is where it began as a local infection and then spread. So you can actually see basically a location where there is more, and then you see that location that the organisms, the infection spread out from that location. Sepsis is toxic inflammatory condition arising from the spread of microbes, especially bacteria or their toxins, from a focus infection. Bacteremia is where you have bacteria in the blood. Septemia is where you have the growth of bacteria in the blood. So they're actually increasing in that environment. Toxemia is where you have toxins in the blood. Viremia is viruses in the blood. A primary infection is an acute infection that causes the initial illness. And then a secondary infection is where an opportunistic organism, opportunistic pathogen came up afterwards because of your primary or predisposing infection. The first infection knocked down your immune system, allowed the secondary organism to take over. Subclinical disease is where there's no noticeable signs or symptoms. It's an inapparent infection. It doesn't actually test for it because it's not, there's not enough of it. Now, when we talk about what makes you susceptible to disease, we have several things that we know. Gender, so a short urethra in females would make you more susceptible to an infection. Inherited traits, an example would be sickle cell, is actually going to make you more susceptible to other diseases. The climate and weather where you live, whether you're tired, your age, how you live, if you're taking certain drugs, if you're going through chemotherapy. The summary is pretty much it's genetics, environmental, and lifestyle. Those are the things that are predisposing factors for your susceptibility to disease. Here we have a graph representing the stages of disease, the disease curve. I want you to notice that it starts out slow, then it goes through a logarithmic growth, flattens out, and then a logarithmic decrease. Now, at the beginning, we have our incubation period, no signs or symptoms. Then we have the prodromal period, and that's where we have mild signs or symptoms. Then we have the actual period of illness. This is where we're going to see an increase in the number of microbes and the most severe signs of symptoms. Then we have the period of decline. The number of microbes are decreasing, signs and symptoms are decreasing over time. And finally, the period of convalescence. These are the stages of disease, and here are the words again for the stages of disease. Now we have reservoirs of infection, places where the infection sets and is passed back to us if we're concerned about us having the infection. If we talk about continual sources of infection, we have for humans HIV, AIDS, gonorrhea, and in these cases, we have carriers that don't actually show the infections. They either have unapparent infections or latent diseases. An example of an animal that is a continual source of infection, rabies and Lyme disease. Some zoonosis may be transmitted to humans. Those are diseases that we find in other animals. Then we have non-living sources of infection. An example of that would be botulism or tetanus. We find them in soil and water. Now here's a selected zoonosis list giving you the disease, the causal agent, and the reservoir. I want you to notice we have birds, we have bats, rodents, we have livestock. A little bit of everything out there are capable of holding on to this disease and then passing it to humans or transmitting it to us. If we talk about how transmission occurs, we have contact, which would be direct or indirect or droplet. So direct, close association, you're touching that person. Indirect would be spread by fomites or inanimate objects. A needle would be a fomite, a doorknob would be a fomite. And then droplet would be transmission via airborne droplets. And you can see here, this is a picture of someone sneezing. The whole point in causing the human body to sneeze is actually to spread the disease. Vehicle transmission would be an inanimate reservoir like food or water. Vectors are arthropods, usually fleas, ticks, and mosquitoes that transmit a disease. Now we have two general methods, mechanical or biological. Mechanical is literally it just physically moved it from one place to another. An example would be an arthropod carries a pathogen on their feet. Biological transmission is where the pathogen actually reproduces in the vector, and then it can be transmitted via bites or feces.